Hello, AP students, and welcome to another JIK presentation. My name is Mr. Salguero, and in this video, I'll be teaching you about cellular respiration. Special thanks to Mrs. Neville and Mr. Simpson who helped me bring this video to life. Also, this video wouldn't be here today without the generous sponsorship of Lavasser Incorporated, teaching biology the way it's supposed to be taught. Let's get started then. Oxidizing glucose with oxygen. As we know, cellular respiration can happen with oxygen and without. So first step is going to be with oxygen. Glycolysis. This is an overview of glycolysis. So basically you have glycolysis, which goes from glucose to pyruvate, and that happens in the cytosol. And then we have the mitochondria, and that has the citric acid cycle and the oxidative phosphorylation. And so basically, the oxidative phosphorylation is an electron transport and chemiosmosis. Electrons carried from glycolysis are NADH, and electrons carried from the citric acid cycle to oxidative phosphorylation is NADH and FADH2. Also, ATP occurs or is produced in each single step, and it's going to be in glycolysis. Only a few ATP is produced, and in the citric acid cycle only a few more, but in phosphorylation is where, in the oxidative phosphorylation is where most of the ATP is produced, which is about 36. So the glycolysis is substrate level phosphorylation, and then oxidative phosphorylation, and that's how it works. Glycolysis. The image to the left is a guide to the most important steps in glycolysis because it is not necessary to memorize each and every chemical step of the process. Below are the net products of the reaction. Glucose, 2-pyruvate, 2H2O, 4 ATP formed, 2 ATP used, 2 ATP, 2 NAD+, 4 electrons, 4 hydrogen ions, 2 NADH, 2 hydrogen ions. Notice the net energy gain indicated in the diagram. Most of the potential energy of the glucose molecule is still in the 2 remaining pyruvates. That will now feed into the citric acid cycle, which is discussed in the next concept. But remember, the net energy gain is expressed in the chemical reactions of the 2 ATP molecules and the 2 NAD molecules. Understanding the reactions taking place. Glycolysis harvests energy for the cell by oxidizing glucose to pyruvate. In glycolysis, which occurs in the cytosol, the breakdown of glucose starts as it is broken down into the 2 pyruvate molecules. The 6-carbon glucose molecule is split into 2 3-carbon sugars through a series of steps. Then step 3 is, over the course of glycolysis, there's an ATP consuming phase and an ATP producing phase. In the consuming phase, 2 ATP molecules are consumed to help destabilize the glucose and make it more reactive. Later in the process, 4 ATP molecules are produced, so in total, glycolysis results in a net gain of 2 ATP. 2 NADH are also produced, which will later be put to use in the electron transport system. After glycolysis occurs, we then move to the citric acid cycle. The citric acid cycle, or Krebs cycle, begins with 2 pyruvate molecules, which is the product of glycolysis. These are derived from 1 molecule of glucose. The first step of this process is arguably either part of the Krebs cycle or just a preparation. But this stage of the citric acid cycle begins with the two pyruvates being oxidized. As a result, a carbon from each pyruvate is lost as carbon dioxide and one NADH is created. This two-carbon molecule is then bound to coenzyme A, an enzyme derived from vitamin B that makes the molecule very reactive. As a result, acetyl-CoA is produced and the Krebs cycle can now begin. Remember, that was just a preparation. The Krebs cycle begins with the binding of acetyl-CoA to a 4-carbon molecule called oxaloacetate. Coenzyme A leaves and a 6-carbon molecule called citrate is made. Citrate goes around the cycle, continuing to oxidize and releasing 2 carbons as carbon dioxide and energy as a product. The remaining molecule is the 4-carbon molecule oxaloacetate, which can be used to repeat the cycle again. Remember, 2 pyruvates are made from 1 glucose, so the Krebs cycle is going to go around twice per molecule. So in total, with 2 pyruvates as the reactants, the product of the citric acid cycle are 2 NADH from the preparation stage, and from the cycle 6 NADH, 6 CO2, 2 ATP, and 2 FADH2 are produced. Here we have a video of how the Krebs cycle works. Electron transport chain. The electron transport chain uses 10 NADH and 2 FADH derived from glycolysis and the Krebs cycle. Electron carriers NADH and FADH2 are moved to the different protein complexes of the electron transport chain that are lined up on the membrane between the matrix and the outer compartment. The protein complexes are able to absorb energy from the electrons, which allow them to push hydrogen ions into the outer compartment against the gradient. The ions will want to achieve equilibrium by pumping back in through ATP synthase, which creates 1 ATP per 1 hydrogen ion entered by spinning and forcing ADP with a phosphate group. So basically ATP synthase is a motor, and the motor works when hydrogen ions come through it, creating ATP. Each NADH will be responsible for the production of 3 ATP, and FADH2 will be responsible for only 2 because of its electron's lower energy level. NADH was split into NAD+, H+, and 2 electrons. When oxygen comes along, it acts as the final electron acceptor and picks up these 2 hydrogens to make H2O, also known as water. On average, the electron transport chain will ideally make 34 ATP, which in addition to the 4 ATP created in glycolysis, and the Krebs cycle gives us about 38 ATP per glucose molecule in cellular respiration. Next, we have anaerobic respiration and fermentation. So anaerobic respiration and fermentation, what anaerobic respiration and fermentation basically is a way to harvest chemical energy without oxygen. Oxidizing glucose has the same first step as oxidizing glucose with oxygen. First, glycolysis has to occur. So glucose right here starts out, goes to glycolysis, and remember the green is the cell membrane. So it happens in glycolysis, and then 2 ATP is produced, a net of 2 ATP is produced in glycolysis, which then forms pyruvate. Oxidizing glucose without oxygen. So there are 2 general mechanisms which cells oxidize organic fuel and generate ATP without the use of oxygen: anaerobic respiration and fermentation. The distinction between the two is that anaerobic respiration has an electron transport chain present, and it only takes place in certain prokaryotic organisms that live in environments without oxygen. Fermentation is a way of harvesting energy without both oxygen and any electron transport chain. There are two types of fermentation. One, we have alcoholic and lactic acid. Lactic acid, an example of lactic acid fermentation would be bacteria and how it uses lactic acid fermentation to create yogurt. And an example of alcoholic fermentation would be yeast and how beer is made. So alcoholic, we start with first. In alcoholic fermentation, pyruvate is converted to ethanol in two steps. First, the release of carbon dioxide from pyruvate. Since CO2 is released, acetaldehyde is created. And then acetaldehyde in step 2 is then reduced by NADH to ethyl alcohol. Now I have a video, a time lapse of how it all works. So basically, glucose is converted to the 3-carbon molecule pyruvate by going through glycolysis. And remember that 2-ATP is produced, not even though it's not is produced, even though I don't have it drawn. Then the pyruvate is converted to acetaldehyde right here, and then to ethyl alcohol. And from going from pyruvate to acetaldehyde, carbon dioxide is released. And so right here, NADH adds an electron and becomes NAD+, adds an electron to NADH, which then loses an electron to ethyl alcohol. Right here, NADH loses an electron to ethyl alcohol, thus going back to NAD+. Fermentation is just an expansion of glycolysis that continuously generates ATP by substrate level phosphorylation, and for this to occur there needs to be a supply of NAD+. So without it losing an electron to ethyl alcohol, we would be stuck here and no more and only 2 ATP would be produced. But since we lose an electron and go back to NAD+, the cycle can start all over again, producing more ATP. Lactic acid fermentation. In lactic acid fermentation, pyruvate is reduced directly by NADH to form lactate. And now we have a video. So basically, glucose is converted, goes through glycolysis, and since it goes through glycolysis, 2 ATP is produced. Then the pyruvate is directly reduced to form lactate. And so basically the same thing that happened before. The NAD+ is adds an electron to make it NADH, and then it loses an electron to lactate to make NAD+, so then the cycle can start over again, producing more ATP. So some organisms with special functions are obligate anaerobes and facultative anaerobes. Obligative anaerobes carry out only fermentation and anaerobic respiration, and in fact cannot survive in the presence of oxygen. Facultative anaerobes are organisms that can make enough ATP that it can survive using either fermentation or respiration. And our last video, aerobic respiration versus fermentation. Aerobic respiration, the final electron acceptor is oxygen, and in fermentation, the final electron acceptor is an organic molecule. Respiration also harvests more energy from each sugar molecule than fermentation does. It actually harvests 19 times more than fermentation does. And this is our last video. So, going back to glycolysis and the citric acid cycle, glycolysis can accept a wide range of carbs. Starch is hydrolysized to glucose, which can then be broken down in the cells by glycolysis and the citric acid cycle. Proteins can be used for fuel